So the first exam question is here. It says describe one difference between the structure of DNA in a prokaryotic cell and the structure of DNA in a eukaryotic cell. Okay, and we've already gone through this. It's an easy one marker. You don't want to be missing easy marks such as this. We know that in prokaryotes, um, the DNA is circular, and in eukaryotes, the DNA is linear. Okay, there was some other example. There was another example that we also looked at. We looked at the point that it was histone bound in eukaryotes, and it's not histone bound, and it's not associated with any proteins in prokaryotic cells. So we just need to put one reason down. I'm just going to say in prokaryotic cells, the DNA is circular, whereas in eukaryotic cells, the DNA is linear. Okay, if I show you the mark scheme, it says in prokaryotes, it's circular, not linear. This is the point that we said. And then you could have had it's not associated with proteins or histones. You could have also said that there are no introns present. Again, this is in a later unit that we cover this. But we talked about this in today's lesson as well. It's not associated with any sort of proteins. And that would get us 1 mark. Okay. The next question is also a 1 marker, and this time it's a tick box one, and it's asking us about the function of ribosomes. So first one is site of transcription catalyzes the joining of amino acids by hydrolysis reactions. So that can't be true. And the reason why that can't be true is because ribosomes are present outside of the nucleus. In the cytoplasm. And because they are present outside the nucleus in the cytoplasm, that means they don't take part in transcription. So any option which is referring to transcription cannot be right. So it's not going to be the first one, and it's not going to be the second one. Okay? It has to be translation. Ribosomes are always involved in translation. They are never involved in transcription. The transcription is literally what happens within the nucleus. The translation is what happens outside the nucleus. And you know that ribosomes are not present in the nucleus, they are present outside the nucleus. So it's either this one or it's this one. Okay, so in terms of ribosomes, site of translation, we got that. Catalyzes the joining of amino acids by condensation reactions, or catalyzes the joining of nucleotides by hydrolysis reactions. Now, nucleotides are joined within the nucleus. So this happens within the nucleus. And because it happens within the nucleus, it can't be this option because we know that ribosomes are not present within the nucleus. They are present outside the nucleus. Okay, so, and they do this exact point. They join amino acids by condensation reactions. So we know that it's going to be this point. It's going to be the third option there. Okay, because they are in the cytoplasm. They catalyze the joining of amino acids, and condensation reactions take place. Okay, and that would be worth 1 mark. Okay, the next question is a 3 marker. So it says scientists investigated the structure of the endoplasmic reticulum. The table shows some of the scientists' results. Okay, so we've got the type of endoplasmic reticulum. We saw in our last lesson that there were 2 types. We've got the rough endoplasmic reticulum and we've got a smooth endoplasmic reticulum. We also looked at a way to learn which one does what. Rough endoplasmic reticulum, because there's a R, we know that it's going to contain ribosomes. Okay, R for rough, R for ribosomes, and therefore it's going to take part in the protein synthesis. Okay, now smooth, we've got S for smooth and S for storage of lipids. Okay, so S for smooth, S for storage. So that's how we remembered it. Now, what does this question tell us? It says use the data in the table to suggest how the structure of the rough endoplasmic reticulum is different from the structure of the smooth endoplasmic reticulum, and how is this related to their functions. So if we have a look at the table, we can see that the rough endoplasmic reticulum has a smaller number of phospholipids. Okay, we can see that from the table. So first thing, we're going to use the data. So we can say that rough okay, we can say that rough has a smaller number of phospholipids. Okay, why is this? It has a smaller number of phospholipids, so what? Okay, well, we know that it doesn't need phospholipids because it's involved in protein synthesis. Okay, it's involved in the production in the proteins. Okay, so it's involved in the production of proteins. Okay, now the smooth, we can see, has a larger number of phospholipids. And that's because it's involved in the storage of lipids. And that's what phospholipids are there to do. Okay, they don't need to synthesize proteins. There needs to be ribosomes present on the rough endoplasmic reticulum. That's why you have a smaller number of phospholipids. On the smooth endoplasmic reticulum, you don't need to produce proteins. You don't need these ribosomes to be present on your surface because these are just present there for the storing of lipids. Or you could say carbohydrates as well. Okay, if we have a look at the mark scheme, it says less phospholipids in rough. So first of all, you need to compare the data, which we did because it says use the data in the table. And then it says rough production or transport of proteins. We said production of proteins. And then smooth production, modification, packaging, transport of lipids, or storage of lipids, storage of carbohydrates, anything like that, which we also said. So we've bagged our 3 marks in that question. Okay. Moving on to a question about viruses. It says, give the 3 structural features found in all virus particles and describe the function of any one of these. So this was a very broad question. You just need to name 3 and you can choose which one you want to talk about. Okay, so all virus particles need to have genetic material. Okay, and the reason why they need the genetic material is because they need to know what to do. They all have a capsid, which is the protein shell of the virus that protects that genetic material. And all of them also have attachment proteins, which we looked at. They attach to receptors, which are present on normal body cells, which allow the virus to enter. Okay, function of one name feature, you can choose any. I'm going to choose capsid because it's the easiest. Capsid is the protein shell which protects the genetic material. Okay, so the genetic material, capsid, and attachment protein, all of which we said, you could have said DNA or RNA, or nucleic acid. Ignore genetic information. Yeah. So we know that the genetic material codes for viral proteins. We know that the capsid protects the genetic material, which is the one that we said, and the attachment proteins bind to receptors which are present on the cell. Okay, again, we said all those points. We've got our 2. Okay, now the next part says explain why viruses are described as acellular and non-living. So we know that they're acellular, and why did we say this? We had a reasoning, and the reasoning that we had was because that it's not made out of cells. Okay, that's the reasoning that we had. So it's acellular because it's not made out of cells. Okay, and then that's acellular, non-living. So I don't think we actually covered non-living, but why is it non-living? What must have, if you're not living, let's say in terms of a human, if a person's not alive, they don't have any processes which are occurring within their body, right? And that's basically what non-living means. Non-living, viruses are described as non-living because they have no metabolic reactions occurring within them. Okay, so no metabolic processes occur within virus. And that's why they are described as non-living. So these two are really important. They're often missed points because they're not explicitly mentioned on the specification. But acellular means that they're not made out of cells, and non-living means think of a normal person. They don't have any chemical reactions taking place in their body. So non-living means no metabolic processes are occurring within that virus. Okay, so no cell surface membrane, or you could have said not made out of any cells. Non-living means they have no metabolism, no metabolic reactions. Okay, so this is our penultimate question. It says give one reason why antibiotics are not effective against viruses. Okay, now the GCSE answer to this would be because viruses live inside cells. Okay, the A level answer to this is a bit different. Okay, remember what I said about bacteria. Antibiotics are used for bacteria. Why are they used for bacteria? They're used for bacteria because bacteria have specific recognition sites. Okay? Bacteria have specific recognition sites. And because they have specific recognition sites, what's an example of a recognition site? Well, it has specific enzymes that the antibiotic can target. It also has a very specific cell wall. It has a murine cell wall. Okay, murine cell wall. And that's what the antibiotics target. They target this murine cell wall. They target these specific enzymes that only bacteria has, and that's how it locates the bacteria. Viruses don't have these specific murine cell walls or specific enzymes. They're literally not even living. So how are you going to target these by antibiotics? So the answer to this would be, I would literally go with this fact because this is a fact that you need to know. Viruses don't have a murine cell wall, therefore cannot be targeted by antibiotics. Okay, so they don't have structural structures or enzymes. They don't have any metabolic processes. They do not have a cell wall or murine. Okay, you could have also said that they don't have ribosomes because that's also something that they can potentially target. Okay, and that would be worth 1 mark. Okay, final question. It says name 2 structures that are found in all bacteria but are not found in plant cells. So what's found in all bacteria? All bacteria must have DNA. So they're all going to have circular DNA. Whereas in plant cells, you're going to have that linear DNA that is histone bound. Okay, they also have a very special cell wall that is made out of a very specific chemical, and that's murine. So they all have a murine cell wall. Okay, plasmids are not present in all bacteria, so don't write plasmids. A flagellum is not present in all bacteria, so don't write flagellum as well. These are your 2 go-to ones. You could have also had 70S ribosomes. 70S ribosomes are also present in bacteria, and they are not found in plant cells. So anything like that. So circular DNA, murine cell wall, or you could have said smaller or 70S ribosomes which are present in the cytoplasm. Finally, it says name 2 features of HIV particles that are not found in bacteria. Do not include attachment protein in your answer. Okay, so ideally with the knowledge that we have right now, there was probably sort of 3 points that we could have put, and that was attachment protein. It says do not include attachment protein in your answer. So we're not going to include attachment protein in our answer. Our next point that we could have said was genetic material. So we're going to write genetic material, which is going to be your RNA or DNA. And what we're also going to write is the protein shell that protects that genetic material, which is the capsid. Okay, those are 3 go-tos right now. You could have also had reverse transcriptase, but again, we'll look at that later on. You could have also said an envelope. Again, we'll look at that later on as well. Okay, so we've said capsid and we've said RNA genome because it says except genetic material or genes. Okay, and these 2 we will come across later. Okay, so 2 marks there and 2 marks there.